Welcome to BioGenerator. I'm Jim McCarter, Senior Managing Director. BioGenerator Ventures creates, builds, and invests in companies that are solving important problems in human health and agriculture. We are the startup arm of BioSTL, a nonprofit that fosters the innovation economy in St. Louis. And over the last 15 years, BioGenerator Ventures has invested nearly $50 million to help start 134 biotech companies here in St. Louis. Those companies have created thousands of jobs and have gone on to raise over $2.8 billion in follow-on investment. We've also supported 170 companies with space here in the BioGenerator labs in the Cortex district, as well as at the Helix Center, the St. Louis County Incubator in 39 North. So for today's Innovators Hour, I am delighted to speak with Dr. Matt Cooper, the co-founder and Chief Scientific Officer of Wugen. Welcome, Matt.
Thank you.
So just to dive in, so now Matt, you grew up in the UK and you studied cancer genetics for your PhD at the University of Surrey.
Yep.
And you mentioned that landing in St. Louis in 2011 for your postdoc was a bit fortuitous, and you were planning on just staying for a short time. And so tell us a little bit about how you ended up at WashU. Why are you still here 14 years later in St. Louis and still working on immunology rather than cancer genetics in particular?
Yeah, well, thanks for having me. It's a great opportunity to share my story and also the story, more importantly, of Wugen and of 007, the cell therapy that we're taking forward. So thank you. So I think my story will resonate with most international students and scholars that have moved to the US. A lot of people will come over, like I did, plan to work for a couple of years, publish as much as they can, and then go back to their home countries. And life has a way of taking over. I met my wife. We have two beautiful children. I got married, bought a house, and as you say, I'm here 14 years later. So moving over and doing science in the US has a way of kind of capturing you, and life takes over and you end up staying in a location. I first came over to St. Louis. I was visiting St. Louis. I sent my CV out to the Division of Oncology, and John DePercio called me into his office for an interview and was telling me about this really exciting project that he has that was about transplantation immunology. And as you mentioned, I was not an immunologist. I had really no interest in immunology. I kind of actively avoided immunology up until that point. But immunology is involved in every aspect of biology. And so I went back to the UK, I thought it over, I decided to take the position, and I did as any good scientist would do. I went on YouTube and I learned immunology. And there's a great lecture series by Harris Goldstein, The Introduction to Immunology, and that's how I got my foot into immunology. And then I moved over to the lab, and I was still a little bit skeptical. I'd heard a lot of horror stories, honestly, about working in the US, people being in competing projects within a lab, and I'm very happy to say that was not true at all. I had such a wonderful time in the lab with John DePercio. He is such a great mentor, and I have very, very fond memories of my time in the lab there. And that's why I'm still here. Then we developed the WuCart 007 and spun out a company, and here we are.
Amazing story. And so to understand your work, we need to kind of go deeper into immunology.
Yeah.
And so there's some key things that those of us who are not trained in immunology kind of need to understand. And so perhaps a place to start is with using the immune system to target cancer. So maybe you can tell us, what is the idea behind CAR T, or chimeric antigen receptor T cell? What is that? And how was it originally deployed to go after B-cell leukemia, as I believe was the first commercialized use of the technology?
Yeah, absolutely. I think to understand CAR Ts, you need to have a little understanding of the function of the immune system and the limitations of the immune system. The immune system has evolved to identify pathogens and infected cells and eliminate them. It's also evolved so that it doesn't recognize self. If the immune system recognized healthy cells, you end up with autoimmunity, and obviously that's bad. So what CAR T cells do is really reprogram the immune system to be able to go after malignant cells, because the immune system isn't going to recognize a cancerous cell unless it has a lot of mutations and it looks different from its normal self. So cancer, being a disease of self, a cell that doesn't know how to die and continually proliferates, looks a lot like a healthy cell. And so that can evade the immune system. So CAR T cells using a chimeric antigen receptor, which is really a Frankenstein molecule of leveraging the targeting capacity of antibodies with the cell killing capacity of T cells into a single molecule, putting that into a T cell and retargeting those T cells against the immune system. As you mentioned, CD19 targeted CAR were the first approved CAR T cell therapies. It's in that system, CD19 is expressed on cancerous leukemic B cells. It's a very robust antigen. And CAR T cells developed against CD19 have been really remarkably efficacious. If you take cells from a patient that has a B cell leukemia, you engineer them with a CAR to reprogram them against CD19, and you put them back into the patient, those cells will seek out and kill the malignant cells with remarkable results. I think that Emily Whitehead is the poster child, the literal poster child of CAR T cell therapy.
Was she one of the early patients?
She was the first pediatric patient treated with CAR T cell therapy. And there's a wonderful video, a very emotional video online, Fight Fire with Fire, that I recommend everybody should go and look at and watch about her story. And 14 years later, she's cancer-free. And that really highlights the impact of cell therapy and the impacts it can have on patients' lives. Because as they describe in the video, Emily was, destined to succumb to disease. These are terrible diseases with very little therapeutic options. And so with the publicity and the press surrounding Emily, that led to a lot of hype and really, got the message out there for the true potential of what CAR T cells could achieve.
And that initial therapy is an autologous approach where you're harvesting the cells from the patient, transforming them and putting them back in the patient,
Exactly.
right? Which is fairly expensive too, to do that on a person by person basis,
Absolutely.
right?
It's personalized medicine.
Yeah.
Yeah. And there have been about 7 approved CAR T cell therapies now by the FDA.
Excellent. So then jumping ahead to around 2017, you and your colleagues at WashU had conceived of a way of using CAR T to target refractory T cell leukemias.
Yeah.
But as you walked me through it, you had to solve like at least 4 problems, right? So this is not just a minimum viable product. This is you have to, if you don't solve all 4 of these problems, you don't have a therapy.
Exactly.
Yeah. So maybe you could walk us through what those were. What problems were you solving?
Yeah, I think with a T cell that you're engineering to target a T cell, clearly those cells look very similar. As I said, a cancerous cell often looks like the healthy cell, so it can evade the immune system. If you're trying to target a cancerous T cell with a T cell, that problem is even worse.
Yes.
I had been to a talk by Jennifer Doudna, and she had really highlighted to me the capacity of gene editing to be able to modify cells. And so if you were to put in a CAR targeting a T cell protein into a T cell, that T cell is going to recognize itself and it's going to kill itself, and you don't end up with a therapy. And so after that talk that I went to with Jennifer Doudna, one of the inventors of CRISPR, I realized that you could use CRISPR-Cas9 gene editing technology to delete and remove the target on the healthy T cell. And that would allow you to create a product that would expand, and you could use that to treat patients with T cell cancers. Now, the other problem, because the healthy T cells and the malignant T cells also look very similar, is if you have a patient that has a T cell cancer, it's going to be really, really difficult, almost impossible, to separate out the cancerous T cells from the healthy T cells to make a product. And of course, if you're layering on CRISPR-Cas9 gene editing to remove the target, you run the risk of removing that target on the malignant cell. And so then if you put that back into the patient, the CAR T cells are going to kill all of the cells that have the target and leave the cancerous cells that don't have the target, and the patient will relapse with a target negative clone. And so other companies at that time have been developing technologies where they've removed the T cell receptor. And the T cell receptor is the receptor that recognizes antigens and leads to a T cell response. If you take T cells from a healthy person and you put them into an immunocompromised person, those T cells are going to recognize the patient in the patient's entirety as foreign. And you'll get graft versus host disease, which is a deadly, life-threatening condition that we obviously want to avoid.
Right.
And so, this bothered me a little bit. You can layer on CRISPR-Cas9 gene editing to remove the T cell receptor as well. And then you could use T cells from healthy donors to create the product to be able to treat patients with T cell malignancies. And so what we developed was a product that was dual edited, deleted in the target CD7 so we could manufacture. It was deleted in the T cell receptor so we could use healthy donors to manufacture this product. And it was then armed with a CD7 targeted CAR that would allow us to be able to target the malignant T cells. There are several advantages of using an allogeneic, so from a healthy donor to create
off the shelf. Right.
One, it's off the shelf, right? You don't have to wait to manufacture this product. It's there ready. A patient comes in, you can give it to them within days, right? You don't have to take their cells, send them off, manufacture through a very long manufacturing process and then send it back to the patients, in which case many patients would have already relapsed and succumbed to disease. This is a very aggressive disease.
Just in that short time.
In that short period of time. And so that's one of the huge advantages. The other advantage is that you can lower the costs. If we can do one manufacturing run and treat 50 patients, then the cost of manufacturing is 1/50th of what it would be.
Right.
And so it makes it much more commercially viable and accessible to take it out to the general public.
Excellent. So you were the first author, and John DiPerzio was the senior author on a paper that was published February of 2018 in Leukemia that provided the proof of concept of the strategy. And I believe it was later that year that you made this critical decision to leave academia. You were an assistant professor, I think, at the time, and pursue this idea full-time as co-founder of Wugen. So, tell us about that decision and why was that such a hard decision?
Yeah, I actually I straddled both positions as CSO and assistant professor for probably 18 months to 2 years. And that was incredibly challenging. I didn't excel at either because, I'm one man, only have a limited amount of time to spend on each position. And so I had to make the decision to either stay in academia or move to biotech. And I was out walking my daughter one Sunday morning, and I was crossing over the road, and there was a car that went through a red light and swerved right in front of me. And I pulled my daughter and her stroller out of the way. Wow. And after that, I had this absolute clarity. I went home and I signed the papers, and I decided to go to biotech. It was an incredibly challenging decision. Part of me knew that Wugen was an incredibly powerful drug, and I wanted to be the custodian of that going forwards. I wanted to make sure that I had a say in how this drug was going to be developed and taken forwards. And had I not done that, I'm not sure that Wugen would be here today. Yeah,
no, I think as the co-founder and inventor, having you there guiding it, is just a critical thing. But you had a lot of help along the way. So, BioGenerator played a role, RiverVest played a role. And so if you could walk us through kind of what were some of the resources that you leaned on and some of the people you pulled in as you and John were building the company.
Yeah, obviously BioGenerator, BioSTL. If there wasn't this opportunity to start a company and house it in the building across the road originally before we moved into here.
And you were in this building for a while.
We were in this building for a while. Yeah. Probably 2 years in this building until we outgrew it. I don't know where, honestly, I don't know where we would have started that company. It would probably have moved out of St. Louis. We would have probably gone to San Diego to some lab space over there, which is a real shame, right? If you create these technologies in St. Louis, it's nice to build the homegrown technologies in St. Louis and foster them here. So there was obviously the facilities, the expertise that provided by the BioGenerator and BioSTL was absolutely pivotal to being able to get Wugen off the ground. And Rivervest and the expertise in company building that was brought from Rivervest was a huge impact in.
And in particular, John McCarter was.
John McCarter.
Yeah, tell us a little bit about John and what he did.
Yeah, John, working with Karen, kind of saw the concept and they got it immediately. They saw the potential of what we were developing and instantly put in some money to be able to prop up the idea and fund this idea to get us off the ground and to where we are today. So that expertise, coming from academia, I have no idea how to build a company, right? And I have no idea how to manage people at that stage. And those are the kind of skills and experience that was brought in by Rivervest and supported by BioSTL to be able to kind of build this company.
Right. And so a cellular therapeutic is not an inexpensive undertaking.
Correct.
So that, we've gotten some companies kind of off the ground with the first $250,000, and then we come in, syndicate to bring in $1 million. But you did a $7 million seed round, an $18 million Series A, a $172 million Series B. Yeah. And so walk us through a little bit about how does that money get deployed, and what are you trying to achieve with those funds?
Yeah, so Wugen had in-licensed an NK program as well, and that was really the driving force behind the $172 million Series B.
Natural killer cells.
Natural killer cells, yeah. And as the markets change and evolve, and as we have developed a drug that clearly, I'm sure we'll get into it, how active this drug is, those funds had to be redirected to make sure that we back the winning horse and get it across the line, particularly in the markets as they are today. So a lot of the early funds went into supporting the phase 1, phase 2 trial for 007, and then for the Series B, that was largely focused on the NK cells.
And as you were recruiting and building the company and bringing in talent, you made a comment to me about how to leverage the experience that people bring from industry. And I think you had some insights into kind of how to leverage that in a way and leverage startup experience.
Yeah, I think I've been thinking a lot about this. There's, I think, a Venn diagram you could almost do of academic experience, biotech, and then big pharma. And I think each of those three circles brings their own talents and their own skills. And it's about being able to draw the expertise out of each of those circles to be able to make biotech work really well. I think academics will be able to see a problem and really drill in and solve a problem like nobody else. But academics need to be kind of kept on track. There's a goal to be able to answer and shiny ideas, which I'm very guilty of, right? I see and I say, I want to research into that, but you have to bring them back, including myself, and focus on the task at hand to be able to get a product across the line. Big pharma, a lot of experience, but that also brings in, I think, a layer of bureaucracy that is not always completely helpful with biotech. And then biotech, the people that really, in my experience, really succeed are those people that are willing to wear multiple hats and change position and roll up their sleeves and get involved like nobody else. So each one of those circles brings their own characteristics and skills that I think is essential for a successful biotech.
Yeah. And so I'm curious to understand more about how you actually manufacture off-the-shelf or allogenic T cells. So, and I know you have a beautiful laboratory across the street.
Yeah.
And you mentioned that the cells are harvested from a single donor. They're used to treat 50 or more individuals. But what does that actually look like in terms of what are the machines involved? What is the process like? How long does it take?
Yeah, it's we manufacture down in Houston, Texas, with a CDMO called Celipon. That's a cell manufacturing facility. It takes 14 days. We harvest cells from healthy donors. They're screened to make sure they don't have any adventitious pathogens that could be transmissible to an immunocompromised individual. So we screen these donors. Those cells are then purified. We take out the T cells. Those are activated so that they start to expand. We gene edit to remove the CD7 and the TRAC using CRISPR-Cas9. and then we deliver a viral vector.
So the cell expansion starts, and then the editing comes in.
Correct.
And then the transfection with the CAR.
Correct. And then we continue to expand those cells out. At the end of the process, we do an additional purification to remove any of the residual TCR positive cells, the T cell receptor positive cells, and those are the cells that could cause that graft versus host disease. So we have a very stringent purification process to remove those. And then those cells are harvested, put into the final fill finish into the drug vial, and then they're frozen. And then they're stored ready to be shipped essentially anywhere in the world to treat patients that have T-cell acute lymphoblastic leukemia.
And so the pivotal trial of those patients is underway with WUCAR 007. Great name, by the way. So I often hear in the media as they're describing clinical trials that they'll say that the only way to test a new treatment is to do a double-blind, randomized, controlled clinical trial or an RCT. But that's different in this case. I think you said that that's not the case for refractory T-cell leukemia and lymphoma. Talk to us a little bit about why isn't it a randomized controlled clinical trial?
Yeah, so the first thing in our phase 1/2 trial in the dose expansion cohort, we had a 91% overall response rate and a 73% composite complete response rate, which is phenomenal, right?
And these are in patients who would otherwise die.
Exactly, right? They're destined to go to hospice, and so these patients have a dramatic response, as we demonstrated in the phase 1/2. And so we've launched the pivotal trial to push forward to get approval. These, there is no other therapy for these patients. So it would be unethical to have a second arm or a blinded arm in which we would give a placebo. It's really not possible to give a placebo, and because there is nothing else available, there is no comparator, right? So
And so what other advice do you have for entrepreneurs, first-time scientific founders like yourself, perhaps another Matt Cooper at WashU, who's an assistant professor who has an idea and is thinking about launching a company? What should they know that you perhaps didn't know 7 years ago?
So I think the first thing would be to, if you have an idea, speak to OTM and get that patented, right? That's the core behind any initial company, is to get some IP to build behind an idea. And
so invention disclosure, and then move on to a provisional, and then full filing, right?
Yep. And then I would encourage them to come and speak to you, and
please do.
to BioGenerator and BioSTL, and get some advice on how to build a company, what is required, some feedback on a pitch deck, for example. Get some contacts on who to reach out to, where to get potential money from, and I would say that once you get beyond that stage and you start to build a company, your people, your staff, your colleagues, that is the most important asset that you have. And so make sure that you have really quality people. If you have any level of toxicity, cut that out straight away. Even if those people are productive, it's much better to have a really collegiate group of people that can really deliver an idea. That's very important. And the loudest person in the room is not always right. That I think is a very important. That make sure that the quieter individuals also get a chance to be heard, because I think that they'll have a lot of really good ideas and advice on how to take a company forward.
How do you structure your meetings now to make sure those voices are heard?
I think meetings these days are a lot smaller. Make sure that everybody gets a chance to be heard. If you have meetings that are too large, then undoubtedly someone's going to take lead in that meeting, and the quieter voices are not going to be listened to.
Mm-hmm. And I'd point out also at WashU Office of Technology Management at OTM, there's now a new ventures group that's headed by Karen, and we should give Karen credit for, it was with Rivervest with Archer.
Absolutely.
And then came over and worked at Wugen.
Yeah.
And so shout out to Karen, and
Absolutely.
that people can connect with her to help within the university getting some coaching on how to build new companies.
Definitely. Great advice.
Yeah. So let's turn to the audience for questions in just a moment, but speak loudly when you ask your questions. I'll try to find everyone, and I'll repeat the questions so that we get them heard by everyone and captured on the recordings. But first, join me in thanking Matt for sharing his incredible story.
Thank you.
What's the size of phase 3? How many sites? What's the cohort?
Yeah, no, so I'll repeat that. And so what is the size of the phase 3 or pivotal trial, number of sites, number of cohorts?
Yep, so technically it'll be a pivotal phase 2 because it doesn't have the additional arm of the placebo-controlled trial. So we're looking to enroll around 100 patients and numerous sites within the US, and then expand into Europe and for a truly global study.
What's the cost per patient?
I'd have to speak to clinical to be able to talk about the cost of patients, but in terms of the cost of drug, that's come down substantially from what we had in our phase 1/2. We spent an awful lot of time optimizing the manufacturing process. In the phase 1, phase 2, we're probably getting about 4 doses from a manufacturing run. Clearly, that's not commercially viable. We spent a lot of time. We now can get up to 50 doses is our target for manufacturing for the pivotal trial. So that substantially drops that cost of goods to really make it a commercially viable process.
How many sites did you say?
In the US, I think we're looking at 7 initially and then expanding out. I mean, these people that have TLL will have historically come to us as much as anything. They really have no other option. There's not many other trials for them to be able to go on to, so they tend to find us pretty effectively. Yeah.
Yes.
What's the ultimate fate of the CAR T cells?
Oh, what's the ultimate fate of the CAR T cells?
Yeah, so upon being an allo product, those will eventually be rejected. So with the CD19 CARs that are autologous, those will persist for years. I think out 10 years, some of these autologous therapies have been shown to persist. With the allogeneic, there's much more immunological pressure for those cells to be rejected, so they don't last anywhere near as long as the autologous therapies. And I think with TLL, the really only truly curative therapy at the moment is to go on to bone marrow transplant. And with a bone marrow transplant, you are going to ablate that patient so that those cells would be lost at that point. So they're not going to persist indefinitely. With a T cell, with a CAR targeting T cells, people don't do very well without T cells for a long period of time, whereas B cells, people can live without B cells indefinitely, but not T cells.
Yes. As you grew and had the option to go anywhere, what kept you in St. Louis, and what turned out to be advantages to our biotech ecosystem that you would not be able to find somewhere else?
That's a great question.
And I'll just repeat it as what kept you in St. Louis is the short version of that. But go ahead, Matt.
I think a number of things. WashU, I think, is a hugely under-tapped resource. I mean, WashU has a world-leading immunology program, one of the best in the world, and people coming out of that program quite often would like to stay in St. Louis. They put down roots, and there's a lot of people coming out of that program that we brought into Wugen and that have excelled at their position. So I think an under-tapped resource of WashU is one. It's relatively inexpensive here as well, right? You get a lot for your money in St. Louis, much more than you would should you be on the coast, and I think that's very important. And I think that, again, I'll shout out back to BioSTL. There's a lot of expertise here and a lot of great ideas and know-how and a really great collaborative spirit, particularly at the BioGenerator, that is a huge resource that really helps us as we move forwards.
Ed.
Yes. Why was the NK license so critical for the company?
Why was the NK license so critical for the company?
At that time, NK cells were seen as the next opportunity beyond CAR T cells. It was seen as the next wave of cell therapy. And I think that there have been certain challenges with NK cells that haven't necessarily delivered on the promise. I think that the asset that we have at Wugen is still very differentiated and would respond very well in cancer patients. But as I say, we have to focus on taking our lead asset forwards, particularly in these difficult times. But that was crucial at that time because people weren't that interested in CAR T cells at that time when we went and got the Series B. But they were very interested in NK cells, and that seems to have kind of shifted back. And what I've learned is the path to success is very rarely straight and narrow. There's a lot of diversions and detours that you take along the way that gets you towards a successful drug.
Why did interest in CAR T wane for a while at that time?
I think there was the perception that CAR T cells would not work in solid tumors, whereas NK cells would. I don't necessarily buy into that. I think that T cells are yet to have their day in solid tumors. There's some great work coming out of WashU and other companies that are going to try and tackle that head on. But I think T cells are yet to have their day.
I know we've got other questions out there. Yes, in the back. Can you speak up a little bit? So question about the U.S. regulatory process and your journey through it, and then expansion into other geographies, in particular Europe?
Yeah, that's a great question. I think making sure you get the right expertise to be able to navigate that system is absolutely imperative. And we have a phenomenal regulatory team at Wugen that has really helped us navigate all of the challenges of going through the FDA and expanding into Europe. We have the RMAT designation and PRIME designation in Europe, and these are for exceptional drug products that get given these designations. It really highlights the large unmet medical need that is addressed by our product. But without the expertise of our regulatory team, then we would not have had any chance of getting them. I mean, they have done a phenomenal job and deserve a huge amount of credit.
Also in the back there.
I'm going to piggyback off that question. At what point did you start engaging in hiring on a regulatory team to start addressing the challenges?
So when did you start hiring that regulatory team?
Very, very early. We, as most startups, rely on consultants at the beginning, and I think that was incredibly important for us. We had an INTRAC meeting, which is a pre-IND meeting with the FDA, and we relied very heavily on regulatory consultants at that stage, and then brought in our own team a little later on to be able to address regulatory challenges.
And I think there someone here had raised their hand earlier. If there was, yes. How does one become a donor?
You would have to speak to the companies that go and source these cells from individuals. They have large repositories of donors that are recallable and get screened in different communities. I think most of ours come from the California area at the moment, but that's led by another organization, and then we purchase these what we call the leukopacks or leukapheresis from those companies.
Other questions? Yes.
What was a diversion that you guys went on that you had to pivot back from, that you learned the most from?
I think the largest diversion, I hate to call it a diversion really, but the NK cells. I think at one stage the company had stopped doing pretty much everything on CAR T cells and was using 99% of resources on the NK cells, right? And it was work that my group was doing in the background, almost kind of secretly, using expired reagents was how we managed to bring this CAR T program back to life. And I think had we not done that and demonstrated that we could lower the cost of goods of manufacturing and make it really commercially viable, then this program wouldn't be where it is today. And so that was definitely a diversion that we took. I learned a lot from that, as I say, that that path is very rarely straight and narrow. So we learned an awful lot in terms of taking a process and modifying it, trying to scale up, overcome a lot of the manufacturing challenges. And then I'm still very hopeful that we'll be able to continue the NK work and demonstrate in solid tumors that we get some level of activity there.
Derek.
So Matt, how did you choose which which disease to attack, and how did you choose the mode of action with CD7?
Yes,
Yeah.
how to choose which disease to go after and the CD7 mode of action.
I think something I've talked about previously is a very influential lab meeting that I had with John DiPercio. John is a phenomenal doctor, very, very compassionate. We had a lab meeting on a Wednesday afternoon, which is our regular slot for lab meeting. And John came in, he had come straight from the clinic. He had a patient that had relapsed refractory TLL, and you could see he was clearly distraught. He had no therapeutic options for this patient, and that patient was destined to die. And he looked across the table with his hand on his head. I was like, Matt, we got to find a way to be able to treat these patients. And that started the conversation of, well, what are good targets? Why CD7? There are other proteins expressed on T cells, but CD7 is a really great target in TLL. It's expressed on virtually all TLLs, whereas other proteins, some are high, some are low. There's variable expression. CD7, it's the CD19 equivalent for B cells, but for T cells. And so that kind of consolidated the CD7 as the target that we were going after. And then I went into the lab and we made some CARs, and it just worked. It's very rare in a scientific career where you have a product and some experiments, and you just know it's going to work. And you do some experiments and you just get fantastic results. I mean, for this one project, I had 9 others that were difficult and didn't want to work and put up obstacles, but everything I did with this project just seemed to work every time. And so that's why I truly had to get behind this product and take it to a company and try and get it out to as many patients as possible. Sometimes you just know. And that's what we felt with WCART 007.
Yes.
So when using CRISPR-Cas9, is genomic stability of the edited cells an issue, and if so, how do you address it?
So with CRISPR-Cas9, the genomic stability of the edited cells.
Yeah, there's clearly always going to be some issues with stability if you're causing double-strand breaks at specific loci. We have done a remarkable amount of work demonstrating that we have little to no off-target activity. And in the patients that we've treated, we haven't seen any issues that have come out from any genomic, as a result of any genomic instability. I think one of the advantages of it being an allo product as well is that it's under a lot more immunological pressure. It's not going to hang around forever. If it was a gene-edited autologous product that would lead to some level of malignant transformation, that's going to be a much bigger issue. We certainly haven't seen anything like that with our product. And I'm not concerned about any level of genomic instability. And I think some of the newer technologies coming out will address some of those issues for future therapies. But I think right now with our product, it works, it gets the job done, and CRISPR is perfectly suited to this application.
Maybe one last question. Yes.
Once and done, or how does that work?
Is it once and done or multiple treatments or?
So the current trial design is to give a single dose, and CAR Ts are a living drug, so you dose it and they expand. It doesn't. They don't have the classical PK of like small molecules or biologics. And so at the moment, it is a once and done. Most of those patients will go on and receive a bone marrow transplantation, which is a path to really a long-term, their only path to a really long-term cure. Having said that, there is the opportunity that if these patients relapse with a CD7 positive cancer again, there's nothing that says we can't give another dose. But we, in the phase of our trials, have not redosed, but we potentially could in the future.
And what's the longest duration tracked patients for?
So the longest duration that we track patients for is well over a year, where these patients are still doing very well. And that's to put it in context of without this therapy, these patients maybe weeks, maybe months, as would be the best outcome.
Yeah, amazing. Matt, thank you for all the amazing work that you and your colleagues are doing, and we wish you all the success. And thanks everyone for joining us.
Thank you.